Can you hear? It's good? Yeah, all good? Okay, thank you. So, good afternoon. So as I was mentioning, Mary Grace joined my lab, and then this is one of the steps for me to get into the graduate program as a member. So thank you to all of you. So, what I'm going to do today is to kind of walk you through in a story way, what do we do in my lab? How potentially, we could collaborate with folks in this center and, do something meaningful science. Hopefully it will work. Okay. So I'm just showing this slide just to give a positive note. The graph shows this 2024 data. You can see on the left, which is cancer incidence, and on the right, it's a cancer-related mortality. And it's a 5-year rate change. What happened in the last 5 years? You could see in the lung and bronchus cancer. Let me see whether I can turn on a pointer. So you can see in the lung and bronchus cancer, thyroid cancer, bladder cancer, the cancer-related incidence is dropping significantly, right, in the last 5 years. And so is cancer-related mortality. But if you look at prostate, the cancer-related incidence went up, but cancer-related prostate cancer mortality went down, okay? So we found more people with the cancer, but many of them survived better, right? Same thing true for breast cancer. And it's very good, in fact, in breast cancer. But in a pancreatic cancer, you see the cancer incidence didn't change much in the last 5 years, but cancer-related mortality went up slightly higher, right? Which means we are not making a good progress in that case. But in overall, if you look at this, in the last 5 years, there's a significant change in the way we take care of cancer patients or the benefit for the cancer patients. How did we do that? Right? There are so many amazing cancer prevention approach, colonoscopy, mammogram, and whatnot. They all making a huge impact in making cancer patients, even if they are diagnosed, to live longer because of the amazing treatment as well as early diagnosis. This is kind of a one-size-fits-all approach which used to be followed for decades or many years, where cancer patient walk in, let's say a breast cancer patient, there is one kind of treatment, and they get some patients benefit from the treatment, some there is no effect. In some patients, you have a lot of side effects. But now, today, with the help of precision oncology, we can stratify the cancer patients into different buckets, and then they get their own respective treatment. This is also another reason why we are able to help cancer patients survive longer, because we have an effective precision medicine for each cancer patient. So here is a classic example, a patient with a BRAF mutation. Is there a way we could dim this light? Or just only the front light? Thank you. That's far better. Thank you. So you can see in this case, it's a patient with a BRAF mutation. It's like a tumor is really bad. And then with the BRAF inhibitor in 15 weeks, the same patient. It's remarkable, right? How targeted therapy could make a difference. But the problem is, while the tumor was eliminated or kind of got significantly reduced, the patient had a tumor relapse. Eventually the patient died. This is a classic example of how targeted therapy or precision oncology, while it's making a difference, we need to constantly learn the tumor as the tumor progresses, so that, okay, now we know we treated the tumor, there is no tumor, but the tumor is coming up. The tumor is no longer depend on BRAF, it's depend on something else. So we need to figure out what it is. Then you can figure out a way to keep this patient alive for long. That's the one problem, tumor relapse, right? The second problem we are dealing with is if someone has let's say prostate cancer, breast cancer, if this is localized, the survival rate, maybe I'll use a pointer here, that way the people on the Zoom also can see, if survival rate with the localized disease is extremely high, like a 95, 98, 99, right? But with the metastasis, the survival rate is extremely low, right? Independent of the cancer type, is prostate, breast, cervical, whatever the cancer it is, once someone has a metastasis, the survival rate is extremely low. So it's extremely important for us to understand the biology of metastasis so that we can also push this red dot towards the right? That's another. So third problem is tumor relapse. Here is a patient who had a basal cell carcinoma of scalp, and this is 20 years later after the surgery, the tumor is popping up. And this year B is 25 years after initial diagnosis, the tumor is kind of relapsed. So again, they're able to do a radiation, able to target this tumor, but the tumor relapse, which is decades later, is a common problem. For example, in ER-positive breast cancer patients, they may have a breast cancer, you treat them, you get rid of the tumor, you kind of decide that this patient is cancer-free, but a decade later, a tumor relapses somewhere, right? These cells sit dormant for decades. So this is a third major problem. So this is a kind of a cartoon of showing that metastasis, tumor relapse, and development of resistance to treatment is the major reason why cancer patients most of the time give up their life, right? They die to it because of these. One factor which connects all the three is cancer stem cells, okay? People have been assessing or kind of figuring out that how the cancer stem cells are generated. One idea was a normal stem cell undergo transformation through whatever process, then you generate cancer stem cells. This is the original hypothesis with which, like, we were all looking, where do the cancer stem cells come from? That is responsible for many of these problems. If we can understand the biology, we would be able to kind of eliminate the cancer stem cells, make these patients live longer. So a while ago, when I was a postdoc, I was just curious to understand the biology of cancer metastasis. While I was looking at it, here is a process, a normal breast tissue on the left. You can see the cells are beautifully organized along the duct, and these cells, which are lining the duct, they are attached with one another through something called a cell-cell adhesion molecule. So that's the way, the fluid from the duct don't leak into the side because this is really tightly organized structure. Now, when a cell like this, a breast cell, get transformed to become a cancer cells, they still continue to express the same cell adhesion molecules. As a result, they are localized. In other words, because these cells do express cell adhesion molecules, which kind of makes them remain local. What I found at the time as a postdoc, there are so many papers now confirming this idea, is that in order for these cells to go from, let's say, breast tissue to the lung, they need to break cell-cell adhesion. They also need break cell matrix interaction. We call this process epithelial to mesenchymal transition. This process happens during embryo development. The first cell which forms upon fertilization is an epiblast, which which forms ectoderm and endoderm. They undergo this process called EMT, epithelial to mesenchymal transition, and generates mesoderm. And mesoderm gives rise to your blood, the heart, the bone, kidney, the ovary. They all come from the mesoderm. So this process normally occurs during embryo development, but in adult, this is mostly silent. In tumor, at some point in tumor progression, they activate this process and then gain migratory potential, okay? And that results in metastasis. In addition, let's say a pathologist looking at a tumor in a lung, and the pathologist, just by looking at a histopathology or the structure they create, they could figure out that whether the given tumor in the lung is coming from breast or prostate or a pancreas or lung or liver, just by looking at a histopathology. In other words, a tumor cell, let's say in this case, breast tumor, they undergo EMT, they leave the primary site, breast tissue, they go to the lung, they recreate a tumor similar to where they came from, in this case, breast. So, which means I was kind of hypothesizing, how is that possible? EMT activates this program or promotes migration, but now the cell undergone an EMT creates a tumor similar to where they come from. As you all know, this is a kind of property of a stem cell. So I hypothesized maybe through this EMT program, cancer cells not only become migratory, but also become a stem cells. That turned out to be true, that in fact, cancer cells, they not only become migratory, but also become stem cells through this process. Okay, this has been proven in many cancer type beyond breast cancer. Originally, we demonstrated this in breast cancer. Now it's turned out to be true in almost most other carcinoma. You can see that, this has got over 10,000 citations in this paper. So what it says is that originally we thought normal stem cells need to undergo transformation, and you want to make a cancer stem cells. But now we know even a normal, even differentiated cells can undergo EMT, and you can make stem cells. This adds a complexity, right? If it's just a cancer stem cells are just simple cell type present in a tumor, you can just find a way to get rid of it. But imagine tumor makes its own stem cells. How will you target it, right? You target a subpopulation and they figure out, okay, fine, there are some stem cells are missing. I'm going to make more stem cells, right? They can make. So we need to understand the process of making, so you can target the process rather than the cells, right? So that is what we are trying to understand. So originally, again, we thought that, there is a direct correlation between activation of EMT, gaining stemness, and the metastatic potential. This was my original idea when we kind of working on this. But we know now the following data which kind of support that. So when we, again, you don't have to wait. If you have any questions, you don't have to wait until the end of the talk. You can stop, we can discuss, and we can keep going. Yeah.
Glad you said that. So in your previous slide, I had a clarification question. So the claim that the tumor has migrated to lung and the cells look very similar to the tumor of origin, is it because now they both are very similar stem-like?
So the question is, I don't know how many people are in Zoom, but you're asking if the cell is leaving the primary tumor and going and creating a tumor similar to where they came from, you have a similar stem cells in both. So it's a very good question. In fact, when the cell at the primary tissue, in this case breast, let's say, they undergo EMT, then they become a stem cells. Once they undergo EMT, then they become a stem cells. When they go to the distant site, they lose the stemness and they create a daughter cells which differentiate and create a breast-like structure. So the stem cell is a transient phenomenon. Yeah.
And it has some notion of memory of its tissue of.
Correct. That's the way we came to know, like if they are coming from a prostate, they create a prostate-like structure. If they come from a lung, they create a lung-like structure in a distant organ. So, so now activation of EMT makes stem-like cells, and which means, and then there is an increase in metastasis when you induce EMT. So if this is true, or the way we asked is it also true in normal stem cells, right? A normal scenario. A postdoc in the Weinberg lab at the time, now he's a professor at Albert Einstein, Wenjin Guo, and I, we took epithelial cells, normal breast epithelial cells, induced them to undergo EMT by expressing snail or twist, one of these factors which can induce EMT. Then we tested them for a stem cell activity. The way we tested that, here is a mouse breast. Here is a 3-week-old mouse. Here is a virgin gland where the entire fat pad is filled with the duct. And then during pregnancy, you start developing this alveoli. And during lactation, they produce a lot of milk and get secreted into the duct. This is a cartoon. Here is a real image. Now, what one could do, you take a 3-week-old mouse, remove the ductal tree, including the nipple. Now the remaining fat pad is enriched with all the growth factor necessary for any stem cell to grow and create a duct. To that, you can inject any cell of interest. If they have a stem cell property, they will create a duct. If they don't have a stem cell property, they will not. Okay. So we took these cells, breast epithelial cells, induce them to undergo EMT. Then we injected them into this cleared fat pad with an idea that I'm making a stem cell, so they should differentiate and create a duct. We spent months and months and months. We couldn't. The reason is, I can ask, if it is a classroom, I would have asked why, but here I'm going to answer. The reason is, when you induce an EMT, when you make them become stem cells, you need to give them the freedom to function as a stem cell. What is the functional definition of a stem cell? They have to proliferate, differentiate, and recreate an organelle structure, whatever they are supposed to. If you induce an EMT, you lock them up as a mesenchymal and stem-like state, they cannot differentiate. So therefore, we could not do it, but we did not understand it at the time. We are breaking our head. But Wen Jun Guo, he continued this work. What he did, he took breast epithelial cells. He overexpressed two factors, slug and SOX9, to induce EMT, but he induced EMT transiently, not stably. Okay? And then he removed the factor which induced EMT, or the DOX in this case, injected 10,000 control cells to this duct. Nothing happened. And these are GFP labeled, so you can look at them in a microscope. And here is 100 cells. I don't know that you can see here. They just created the entire ductal tree, including basal cells, luminal cells, everything you can think of. So all you need is a reversible activation of EMT is necessary to create metastasis or to behave like a stem cell, not stable activation of EMT. Are we all on the same page? Okay. So now coming back to metastasis, you need to induce an EMT to make stem-like cells, but you cannot lock them up in the mesenchymal state, right? It's a reversible process. So that is where, so you need to induce an EMT. There is a reversibility at some point. If you look in the top, the cells do retain the reversibility until this point. And then once you go to the other side, once they lose the reversibility, they cannot develop metastasis. The metastatic potential goes down when they lose the reversibility. Yeah? Please stop me again. We don't, I don't want to talk only, just continuously talk. It'll be nice send slide. So now, the couple of review articles, in case any of you wanted to kind of understand, there's one is called vimentin, cytokeratin, good, alone, bad. Together, we talk a little bit of these ideas. And there's also proactive and reactive roles of TJ-beta. We talk about this reversibility a little bit on that. So we know cancer stem cells are bad. Now you can make more cancer stem cells by activating EMT. And we also know reversible activation of EMT is necessary, not stable activation of EMT. Yeah? Okay. So now what I'm going to do in the next several minutes, I'm going to talk about how can we use this information to figure out a way to address the three problems I listed earlier, right? The three problems I articulated is tumor relapse, metastasis, and treatment resistance. How can we address those problems? How can we go back and figure out a way to help these cancer patients? So next few slides, I will show you how are we approaching this problem from a tumor cell intrinsic manner. What I mean by that is that if you understand how the carcinoma cell, just a tumor cell, get transformed or undergo EMT from a tumor cell point of view, then you can go and intervene that, or you can detect that. Most of the time, the reason I'm saying is that in a tumor, it's not just made up tumor cells. You have so many other cell types. So here, I'm going to show you what we are trying to do from a tumor cell point of view. So the tumor cells, when I say EMT, I just said one word, EMT. But EMT is not a simple one factor goes and induce an EMT is done. It's so many things can regulate EMT. There is a hypoxia, cytokines, ECM, CAPS, even a chemotherapy. And they all can activate various pathways that can induce various transcription factors. Then you end up in EMT, leads to all this. Now, so if you say patient A has a hypoxia-induced EMT, patient B has a ECM-induced EMT, how do we target them? Unless otherwise you know how the EMT was induced. The second approach you can also take, if everything is feeding into something, right? If you can find something which is downstream of all this, if you can find that, then you can go and intervene that with the hope that you can block all the things which are inducing EMT. I always believe I'm very lucky. So we were very lucky to find one such factor. Here, Brett Hollier, who was a postdoc in my lab, he looked at how this transcription factor, one of the factor called FOXC2, which you can see is not expressed in most adult tissues except in adipose tissue. Yeah. This is an adult various organ somebody used is by northern blot. By looking at an RNA, it's hardly present. Here is a breast epithelial cell induced to undergo EMT by expressing any one of these factors. And here is the control cells, which says vector. And we are looking at a E cadherin, which is one of the marker of epithelial cells. When you induce an EMT, they lose E cadherin. They all gain vimentin, just nothing surprising. It's all known in the literature. What we found, or what he found, is that FOXC2 get induced when the cell undergo EMT, independent of the method he used. Okay, he tried many methods. I'm just showing you some from the paper. That when you induce an EMT, or even if you isolate stem cells from a tumor, they tend to express higher level of FOXC2. So we thought maybe FOXC2 is functioning downstream of these various EMT inducing signals. If you can intervene the FOXC2, you would be able to target these cells undergone an EMT. So he used shRNA, and you can see in this cell model that he was able to reduce some of the expression of these EMT-associated markers. Most importantly, in the right, you can see just by inhibiting FOXC2, he was able to reduce invasive property of these cells. You can see in this image here also in the vimentin, suggesting that one could target FOXC2. But FOXC2 is a transcription factor. How will you target? It's now we have a method of using a PROTAC, but still it's not ready, right? How do we do it? To reiterate that FOXC2 is an important player, here is a image by Maria where one stem cell is dividing and giving rise to two daughter cells, where one daughter cell is expressing FOXC2 and another daughter cell is expressing a factor called a NUM. The NUM is related to differentiation, and FOXC2 is related to stemness. Okay, in the asymmetrically dividing cell, or one mother cell is giving a two daughter cell. One daughter cell is going to go on to become a stem cell. Another one is going to be a differentiated cell. All this decision is happening at the time of cell division of few hours, right? Again, reiterating that FOXC2 segregates with the non or undifferentiated cell, and FOXC2 get induced following EMT, suggesting that there is a tight connection between stemness, EMT, and FOXC2. Okay. So what another postdoc of mine, instead of using shRNA, he said, can I use some small molecule, right? How would you just randomly choose a small molecule? So he said, I'm going to look at a FOXC2 protein and look for any potential site for phosphorylation by any one of the kinase. And he found a site for a p38 MAP kinase, a highly conserved site for a p38 MAP kinase, which you can see. And then he also did a Western blot of these cells induced to undergo EMT. You can see when they express FOXC2, they also had a phosphorylated form of this p38 MAP kinase. You see this cell, the first one, vector expressing cells, there is no FOXC2, there is no phosphorylated form of MAP kinase. So he hypothesized that there is a tight correlation between phosphorylated form of this MAP kinase and FOXC2. And this in the phospho MAP kinase has a phosphorylation site in FOXC2. There could be some connection. So with that, he went and exposed these cells with this p38 inhibitor, and he saw just by exposing them, he was able to get rid of the FOXC2 protein. So somehow, phosphorylation of FOXC2 by p38 MAP kinase makes them stable. Okay, if you get rid of the p38, you don't have a FOXC2. So we just amazing tool we found. So one of the things we always try to do is, if you take one cell line, you do some experiment, you get an amazing data, and it cannot be reproduced even to the next cell line, it's useless. So we want to do in a multiple cell model. And that's what he has done here. And most importantly, here is something called a spear assay. So the spear assay is, you take cells and you plate them in a suspension culture, okay? Most of the differentiated cells will die, but stem cells find a way to survive in suspension. They proliferate and they differentiate and create a colony like this. In this colony, you will have approximately 300 cells, okay? They secrete their own ECM. They are growing in suspension. This is the way you can assess whether a given cell is a stem cell or a differentiated cell. And we also do this experiment in the presence of some chemical called methyl cellulose. Methyl cellulose prevents cell aggregation, so they have to grow independent. So any cell grown in that colony coming from a single cell. Okay. By doing this, he found that he not only was able to inhibit FOXC2 expression, but also the spear formation, which correlates with stemness. So he was able to inhibit stemness. Now, going back to in vivo study, he treated the mice having tumors with the p38 inhibitor. He did not see any change in primary tumor growth, simply because he's affecting stem cells, which are 1 or 2% of a tumor, not the whole tumor, okay? And with that, here is a primary tumor. There's no difference in a primary tumor volume. But when you look for metastasis, these are luciferase labeled. Here is a vehicle-treated group, and everyone had a metastasis. When we study metastasis, we don't inject the cells in tail vein. We put them in a mouse breast, ask them whether they will go to the lung, okay? And here is a metastasis from the breast tissue to the lung. Every mouse had a metastasis to the lung, but when he treated them with the p38 inhibitor, none of them had a metastasis. Another thing we also make sure to do, we use an immune competent mouse model, which means they have an intact immune system, rather than using an immunocompromised mouse model, which what many people use. So we have a new postdoc in the lab. I wouldn't say new, he has been working for a few years now. He's developing that small molecule further into now he's able to show that by combining that with the chemotherapy, he can not only get rid of the metastasis, but also primary tumor. Okay, we are finishing up that story on that. So in addition to that, we also look for other small molecule which could target FOXC2 by doing a small molecule screen at Scripps Research Institute in California. This undergrad, he was an undergrad at the time. His name is Mike Bolong. He screened some million compounds to start in a high throughput screen. But then later he started out to some 50,000 compounds, and he pulled out one compound having a high toxicity to cells expressing FOXC2. And when he looked for how this small molecule works, he found it binds to the protein which I was talking earlier, the vimentin. Okay, and so based on this, he kind of exposed the cells to this 5-1 for some whatever reason. Only in stem cells, this 5-1 makes the cells divide, but they get locked up in a multinucleated state. In other words, the stem cells, they divide and they don't become a daughter cells. They just locked up in one cell with a multiple nuclei. So this is with a small molecule. Then he said, why don't we make a possible mimetic? We know it interacts with the vimentin. So he made a mutant form of vimentin. He put it in a cell to test is the molecular mechanism is similar. And he found that in fact, even expressing the vimentin in these epithelial cells induce them to undergo this multinucleation. So we know how the process is working. Please, again, stop me if it's like, beyond something which you would like to discuss any of these. So Nick Kubridge, who's sitting here in the back, so he said, can I elaborate this further into more model? So he took many cell lines and then he kind of assessed their ability to form the spear, which I was mentioning earlier. And you see these cells form a spear. And then he said, if I expose these cells to this compound or the mutant vimentin, he saw multinucleation only in cells which has stem cells. In other words, the cells which are becoming multinucleated in response to this drug or a vimentin turned out to be stem cells. Okay, so that's our goal, right? We want at the end of the day, we want to eliminate the stem cells. Particularly the process of making up stem cells is what we want to target. So here is a beautiful data where he took this mutant or the phosphomimetic mutant of vimentin. He expressed in epithelial cells. Nothing happened. There is no multinucleation you can see here. And then this is another epithelial cells. Nothing happened. But if he takes the cells which has undergone an EMT, the cells become multinucleated. Okay. So in other words, if you take a fibroblast which also express vimentin, this compound does nothing. But if you take epithelial cells, which is not undergone an EMT, this compound does nothing. Only when a cell undergone an EMT, this compound or this vimentin kind of mess with their behavior. So based on that, he said, okay, I'm going to take these cells, which has stem cells or cells undergone an EMT and a cancer cells, expose them to this compound, push them to become multinucleated. Then I'm going to take those cells, pass them through the fax machine. In a fax machine, you can eliminate all the cells which has undergone or which become multinucleated, right? You eliminate it and take the remaining cells and then you inject them to the mice. If these multinucleated cells are responsible for making metastasis, then there shouldn't be any metastasis, right? It's a beautiful experiment, and you can see that in this case, you can see the control. Maybe I can zoom in. You can see better. And you can see this white structures are the metastatic nodules. And you have a lot of metastasis in the control. But the minute you eliminate those multinucleated cells, inject same number of cells to the mice, and you see there is no metastasis, indicating those cells which form metastasis are the stem cells, and those stem cells are becoming multinucleated, and he eliminated them through fax analysis. Yeah. So now coming back to the story again, you are welcome to review this paper, which has a lot of information relating to this. Now coming to, so in one side of the lab, we are trying to figure out how can we target these cell population, particularly by targeting the process. The other side is, can you identify them, right? It's extremely important. If you look at a tumor tissue under the microscope, everything looks the same. To me, I cannot differentiate anything under the microscope. Here, if somebody has done a staining, the cytokeratin is for epithelial cells and vimentin for mesenchymal cells. You see this tissue, tumor tissue has epithelial cells and mesenchymal cells. Both are not intertwined, but both not expressing the same marker. In other words, there is no cells undergone an EMT on the left. But if you look on the right, you do the same thing. Again, epithelial marker and a mesenchymal marker, there are a lot of yellow cells, indicating that these cells have undergone an EMT. They are also present. How much they've undergone an EMT? Are they in a reversible state or irreversible state? No idea. How do we find out? And is every cell undergone an EMT is a bad cell? Or maybe some cells, one of the way we were kind of puzzled is that if you take 1,000 cells induced to undergo EMT, and you plate them on the spear assay, which I was mentioning earlier. Spear assay assess for stem cells, right? If you take control cells, 1,000 cells, you get 2 or 3 spear, indicating you have 2 or 3 stem cells out of 1,000 cells. If you induce an EMT, and we know they become stem cells, now it means if you take a 1,000 cell, you should get a 1,000 spear if every cell undergone an EMT become a stem cells. But you get only 50, maybe 100, right? You don't get 1,000, which means you are making stem cell, but not every cell, the product of EMT, not every product of EMT are stem cells. And which means in this case, there are some cells maybe a bad cell and some cells are good cells, not good cells. Some cells are doing nothing there, right? If we can differentiate that, it'll be big help. How do we do that? So this is a study of Petra Denhelander, who's also in the back. So you take an epithelial cell, you induce them to undergo EMT, you generate mesenchymal looking cells. They are not fibroblasts, which are totally coming from a different lineage. And you also generate mesenchymal looking cells with the stemness. This is an idea, right? How do we test that? So she took an epithelial cell, induced them to undergo EMT. They become mesenchymal. You can see from here from left to right. You can see more carbaloid structure to become a single cell. And you remove the TJ beta 1, they revert back. Okay. Now here is a Western blot looking for protein markers. Fibronectin, for example, it goes up and goes back down once you remove the EMT signal. Nice. And here is a spear formation, which I just described. The spear formation goes up as the time goes. And then when you withdraw the TGF-beta1, it goes back. Here is another marker, again goes up and goes down, stem cell marker. Now, if you continue to expose them to TGF-beta1 for, let's say, for 15 days, fibronectin goes up, spear formation goes up until day 6, and then stable, right? It didn't go up much. So indicating that the stemness takes a while for it to go up, and then at some point it's kind of saturated or reached an equilibrium. But if you look at a mesenchymal marker in 18 hours, you start seeing a mesenchymal marker. And you calculate the number of the doubling time, how many hours it takes for the cell to divide from 1 to 2. It takes approximately 22 to 25 hours, okay, for these cells to divide to give rise to 2 daughter cells. But within 18 hours, you are able to see vimentin, fibronectin. It takes several days for it to reach stemness. So suggesting that when you induce an EMT, the mesenchymal program, which is kind of signal transduction mediated, and you can have the phenomenon happening in no time. But for stem cell property to come, it takes multiple generation or population doubling before it can become a stem cells, which means do they, or the activation of EMT in a cell division dependent manner, reprogram the genome and give rise to stem cells. But for a mesenchymal program, you don't have to divide. You don't have to reprogram the genome. You add the TGF-beta1, boom, 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 some SMAD dependent signal transition pathway, you upregulate mesenchymal marker, right? So if this is true, what Petra said, I'm going to block cell division and then induce them with the TGF-beta1. That's what she did here. Here is the thymidine, which blocks cell division, again 6 days. In the control cells, you can see they all upregulate fibronectin, and they also upregulate the spear formation, you can see in the bottom. But when you block cell division with the thymidine, they still upregulate fibronectin, N-category, and all that. But look, the spear formation is none. So indicating that in order to gain mesenchymal program, you don't have to divide. You can just to take a cells, expose them, you get a mesenchymal program. But to become a stem cells, you need to divide, right? So this kind of provided us a nice handle to separate cells which can become a mesenchymal cells, which can become a mesenchymal, or which can become a stem cells. And we always want to make sure what you find with thymidine is also true with other cell cycle block, and that's what she found. Now, we did a single cell RNA, in this case, total RNA sequencing, and we found that if you block cell division, you end up in making mesenchymal cells. But if you don't block, you become the pink. These ones are stem-like cells. These are mesenchymal cells. You generate both. Okay. We also had a single cell RNA sequencing data. With that, we kind of mapped these gene signature, which we derived from this analysis. And what we found is that if you have a mesenchymal gene set or gene signature, and you map that on stem cells, the other way around. So here is a stem cell, cells are isolated for stem cells, and every stem cell gene expressing cell are also mesenchymal. But not all the mesenchymal marker expressing cells are stem cells, only subpopulation. Okay, so in other words, every stem cell express mesenchymal marker, but not every mesenchymal looking cells express stem cell markers. Are we all clear on this? Yeah. And most importantly, the stem cell signature is able to do a clinical prediction better than the mesenchymal signature. So now, how does it do? How does this cell division dependent regulation of stem cell program functions or operates at the molecular level? So when we looked at the RNA sequencing, one other thing Petra noticed is a high change in spliceosome and mRNA splicing, which is kind of significantly affected when you expose them to cell division block. Okay. So by looking at various genes, I'm not going to tell you kind of walk you through exactly how it is. But then she found one protein called ESRP1, which is called epithelial specific splice protein 1. Okay. And you can see when you expose the cells to TGF-beta1, this protein goes down. And when you withdraw the TGF-beta1, this protein comes back on. It goes down and comes back on. And if you block cell division, it doesn't go down at all. So indicating there is a correlation with the ESRP1 and gaining stemness. This is like, again, I don't believe in just taking one cell model and figuring it out that, we found the solution to the biology. You want to go beyond one cell model. So she took multiple cell model and then asked, is there a relation between ESRP1 and a Vimentin? Vimentin is a mesenchymal marker. ESRP1 is an epithelial marker. And you can see there's a direct inverse correlation between ESRP1 expression and a Vimentin expression, which means when a cell undergo EMT, they lose ESRP1. Okay. Again, the way ESRP1 works is that it regulates Notch signaling. Okay, Notch signaling is related to stemness, which has been very well established in the literature. So she overexpressed ESRP1 in these cells instead of thymidine, instead of blocking a cell division. She overexpressed ESRP1 and treated them with the TGF-beta1. If you overexpress ESRP1, fibronectin still goes up. There's no impact on mesenchymal program. But if you look at a stem cell program, the TGF-beta1 was unable to induce stem cell program. So it's basically correlating with our original observation that you need, in a cell division dependent manner, the ESRP1 goes down, which allows the cells to become a stem cells. But if you don't allow the cells to lose the ESRP1 by forcefully expressing ESRP1, and then you still expose them to TGF-beta1, their ability to gain mesenchymal marker is not affected, but their ability to become a stem cells is completely inhibited. So this is kind of an operating in a Notch-dependent manner, which I'm not going to go into, but in a summary, in a differentiated cells, you have a ESRP1, which inhibits Notch, okay? But in the stem cells, the FOXC2 transcription factor I was talking about inhibits ESRP1 by some mechanism. Once you inhibit ESRP1, you activate Notch, and the Notch provides stemness. Okay, this paper just got accepted in the journal called Eye Science. So since it's a kind of a computational biology, I thought I will also show a little bit of data, what we have done in a computational biology. Right. Here is Herbie Levin, who is my good friend, also long-time collaborator, and he's a National Academy member. He's at Northeastern University. So Herbie and we have been curious to understand what happens at a single cell level when a cell undergo EMT, right? So we took epithelial cells, induced them to undergo EMT, and then here is the clustering of these cells induced to undergo EMT. This is 6 days of exposure or 8 days of exposure. And then they have done a pseudo-time clustering. So here is the pseudo-time analysis. And then you could clearly see the topmost pathway is EMT. Of course, we are inducing an EMT. It should be in the top. And we are inducing EMT with the TGF-beta1, which is the second. And the beta-catenin signaling is stem cell pathway. The Notch pathway, which I was telling you, we discovered in a different way. The Notch pathway is also high, HOG, OXPAS, whatever, right? So then when you take the pseudo-time clustering and look for expression of, let's say, microRNA, and you see the microRNA 217, which goes down in a pseudo-time dependent manner in this case, and the associated target, when this microRNA goes down, their target goes up, because I'm sure you all know the microRNA binds to predominantly 3' UTR of mRNA. It degrades the expression. Now, if you don't have the microRNA, the gene expression goes up. And many of these microRNA and their associated targets, you can see they're going up when you lose the microRNA. Okay. The interesting experiment, what they did, this is kind of very fascinating, which I have no idea how they did. I'm sure you all will understand this better than I do. So he did this systemic inhibition of EMT signaling computationally. Now, we spend kind of days and I cannot explain how much time we spent to figure out each one of them. They spent like few days and they came up with this model. Okay. The model is cell-cell communication. Right? The one in red is control, the one in blue is a pathway repression, and cell-cell communication, insulin signaling, hippo signaling, these are various pathways. And here you can see clearly, if you inhibit YAP signaling, which is one of the pathway induced in this process, he said there is no impact on the YAP signaling, the YAP pathway is inhibited, but nothing happened to Notch, nothing happened to TJ-beta. When he inhibited Notch, he said the YAP signaling is affected, and the Notch signaling is affected because you are inhibiting Notch, and so is the TJ-beta signaling. Okay, when you inhibit TJ-beta1 signaling, because here you are adding TJ-beta1 to force the cells to undergo EMT, and if you inhibit TJ-beta, of course, the YAP-TAZ is gone, and Notch is gone, and also TJ-beta. So based on this, he hypothesized the dominant signaling which operates in this program to control the stemness is a Notch pathway. Okay, that's a paper we published. Again, he's the master of these things. I am just a messenger here. So now coming back to the last set of data, as I was mentioning earlier, when you look at a tissue under the microscope, you have no idea what is what. But somebody can draw a cartoon and say you have a cancer stem cells, you have a CD8 cells, T cells, regulatory T cells, and whatnot. But I have no idea which is what in that, right? So we need to understand that. So what happened in tumor progression is that if you think about a tumor at an early stage, the immune microenvironment tend to be tumor killing, okay? But then at some point, the immune microenvironment switches from tumor killing to immune suppressive and tumor promoting, right? So one of my postdoc, Rubia Joseph, she looked at this problem beautifully. She used 2 cell model. She put them in a mouse breast, and one cell model goes to the lung, another one just stays low, okay? And then the question is why this cell is able to go to the lung and these cells are not? Now, is it because these cells are unable to get into the circulation? If I allow them or force them to get these cells into the circulation, can they develop metastasis? So then she injected these cells into the tail vein of the mice. Then now you are helping them to get into the blood. Now, can they develop metastasis? Yes, they can develop metastasis, which means the limiting factor is one cell is able to get into the blood easily. Another one is not able to get into the blood easily, okay? Who controls it? Is it tumor cell intrinsic or tumor cell extrinsic? And it's difficult to test a tumor cell intrinsic in this process, but easy to test a tumor cell extrinsic. There are so many mouse models out there where you can test simple immune cell function. This is a not-skid mice, and same all in a biopsy background, not-skid mice. They don't have T cells, B cells, and NK cells, okay? You take these same 2 cells, put them in a mouse breast. In the absence of those 3 cells, the first cell, which was failed to get into the blood, now is able to get into the blood, is able to go to the lungs, which means one of these 3 cells is controlling this process. Then he said, okay, can we narrow it down? There's a nude mice. They don't have a T cells, right? You put them in a mouse breast, it goes to the lung. So which means it's a T cells among the 3. And then within the T cells, there are 2 subsets. It's called a CD4 and CD8. And in a CD4 knockout, where there's no CD4, nothing happened. It's really the cells not able to leave the primary site. But in the absence of CD8, these cells not only left the primary tumor and also developed metastasis in the lung. So the conclusion was it's a CD8 T cells which is controlling the tumor cells from leaving the primary site. So I want to show you this beautiful data where then you isolate tumor cell educated T cell, right? The T cells, you cannot just isolate T cells from a peripheral blood and ask the T cells to kill a tumor cells. They have to educate and get exposed to the T cell antigen and the tumor cells. She isolated T cells from the mouse tumor and labeled them in blue. She also isolated tumor cells and labeled them in red. And here is a non-metastatic tumor where the tumor cells were not able to get into the blood and can develop a metastasis. Here is a metastatic tumor. Now you see on the left, the T cell will engage with the tumor cell. If the T cell is managed to kill the tumor cell will turn green because there's a dye which you can add to a dying cell, and So the tumor cell is dead. Okay. So, but if you do the same experiment, in the other case, the T cell, this is, they try their best with so much of trial. At the end, the cell which die is a T cell, not the tumor cell. Okay. So we have done this extensively, and what we found is that, I'm just going to summarize the data in the slide, that it's in a non-metastatic scenario, it's a CD8 T cell which is kind of preventing the metastasis. But in a metastatic scenario, there's a platelets which get into the tumor by some means. We don't know how. They produce a hormone called CXCL, chemokine called CXCL4, which converts something monocytes to MDSC, which inhibits CD8 T cells. Once the CD8 T cells are inhibited, now the tumor cells are free to go and develop metastasis. We have, the whole thing is in that paper. Again, I'm not detailing the story. Again, coming back to this problem, that how can we address the tumor relapse? This is where, we all can work together if any of you are interested. So the model is, you have a tumor cell intrinsic resistance or acquired resistance. When I say intrinsic resistance, imagine you give a BRAF inhibitor. There are some clones don't express BRAF mutation. Now they are not going to respond. And they may have a different mutation, and they may come eventually clonal expansion and create a tumor. Or acquired resistance, in response to the treatment, some cells may gain mutation, and then that will lead into new clones emerging and develop metastasis. So here is the kind of a review article, it's not ours, showing that, non-functional passenger mutation, they just carry along. But if there's a functional driver mutation, in other words, the mutation gives a benefit to the cell, now those clones will expand, right? So if the beneficial, there are many ways these clones can get a benefit. It could be epigenetic phenomenon, it could be point mutation, or if this point mutation give rise to a new antigen, then those cells will be eaten and get eliminated. But if the mutation is kind of a subclonal point mutation, is more of a not a new antigen producing a mutation, then those clones will expand because immune cells cannot target. So is the epigenetic alteration, they will not produce new antigen, right? So they will also expand. So it's very important for us to understand the dynamics. How do we do that? So we have some of these fancy equipments. Was fortunate when I moved here. There is a foundation here. They gave like a $5 million to study various things. Plus, Brown was, the money, startup money, and with all that, we bought these fancy equipment, cost around over $500,000. But what it does is that, those of you who may not know, is that in a Chromium, it allows you to map every single cell to sequence. The Visium allows you to map them spatially, but not at a cellular level, at a few cell level. The Visiums, Chromium, Visium, Xenium. Xenium allows you to map every cell at a subcellular level. Okay. This is, I think, image created by either Mary Grace or the RA who was there in the lab before, showing that that's like a graining various fruits and making a juice and figuring out what is there in that. And this one is kind of mapping at the spatially, but here you can go at the subcellular location. Okay. So what Mary Grace is doing in the lab is, now we have, again at Brown, we have a number of breast tissues where these are pre-treatment biopsy. We got like 12 patients. And these patients have never received any treatment yet at this time of biopsy. We got those tissue. We have done a single cell RNA sequencing. We actually put them in all these platforms. Then the same patient got a treatment. We know 4 patients, the tumor vanished. There is no tumor. Okay. In another 4 patients, the tumor didn't disappear at all. It's just intact, which means the primary tumor, treatment naive, where the cells are carrying an information whether the patient will respond or will not respond, right? What Mary Grace is trying to figure out is that, can I identify genes, pathways, cells, which could decide whether the given patient is going to develop resistance or whether given patient will become respond to the treatment. This is a chemotherapy, but we can do similar things for any treatment. Right? At this point, it's very expensive. That's why we are just starting with this process to start. This is some of her analysis where she showed this is some genes she used to map different cell types. Here is a UMAP analysis. And just to give one example, in a resistant population, a gene called VTCN1, which is highly expressed related to the patients who are sensitive, right? And this gene has been associated with similar process in other papers. So we are trying to use this to create a small gene set, which we can map it back to xenium and potentially create a gene panel. And then if you have a 13th patient walking into the clinic, and the idea is you can run them on a xenium and then say, this patient has a very high chance will respond to chemotherapy or will not. If they will not, can we provide an alternative suggestion, right? So in summary, sit up on time. So treatment resistance is driven by tumor plasticity, which can be multiple process, epigenetic, mutational, all that. And current methods lack resolution to predict these resistance at the single cell level. And the most importantly, remodeling of the tumor microenvironment through EMT also extends the plasticity beyond tumor cells because these cells, they function on modulate the behavior of the other cells. So we need to do single cell analysis and identify novel actionable biomarker. Identifying a biomarker is easy, but that biomarker is not going to provide a solution. So you want to have an actionable biomarker. When I say actionable, a drug target, right? Then you can go and target that biomarker. Simulating therapeutic strategies in silico. I'm sure you all know there's something called a Gemini, which is not your Google Gemini. There's a Gemini, which is an in silico digital twin. It's like you replicate all the data in a digital scenario and run parameters to figure out whether some patient will respond or not, right? That's where things are going. As I mentioned, your tumor cells are undergoing EMT. They are communicating with this microenvironment. We need to understand this communication and understand what the tumor cell is doing. It's a complex process. We need an army of people working on this. So these are all the beautiful people from my lab, and they're all addressing different. I didn't get a chance to speak everyone's data because in the interest of time. So really, many of them are here. Thank you to all of you for coming here. And this is, it's Wafik, who's the cancer center director, was kind of responsible for me moving to Brown. Here's again another amazing picture. And thank you. This is the funding support again. There's various agencies. We got funds to do many of these work. Thank you. I guess everyone understood everything I said.
That clear.
Maybe we should have an exam. Anyway, yeah, please.
Broad question, but you're coming at the problem from different angles and focusing on one transcription factor, but the problem seems to be multidimensional, multi-scale, lot of factors, lot of environment playing the role. So are you thinking about that aspect of it, like just the scale of it?
Oh, absolutely. That's why, while the EMT can be induced by multiple methods, so we got lucky, we found one factor which kind of falls in the bottom of all those pathways. Is it enough? Probably not. Should we do more? Yes. Can I do more? No. So my bandwidth is, whatever the Money Lab bandwidth is, only so much we can do. But ideally, you are absolutely right. There are so many people, when there's a group, I don't want to say who they are. There's one guy, I have been trying to teach that EMT is important for metastasis. It's a clear evidence. It's not that I'm trying to make it up. There's one other person who has been saying EMT is not important for metastasis, right? And we both used to teach the same class. at MD Anderson, and I go and teach that EMT is important. The students were sitting and listening, and then he goes the next class is EMT is not important for metastasis, right? It's finally now everybody's on the same page. Now even that person agrees that EMT is important for metastasis. So it's unfortunate that it's not that what I think is right or important. What we all think is important. So which means everybody has to get caught on to the similar idea if what I'm proposing is right. If I'm wrong, I'm wrong. It's fine. But if it is right, it takes time it took so many years for people to accept that you can use the word cancer stem cells. Okay. When we published it, they said, don't put stem cells, put stem-like cells. That was a recommendation from the cell editor. She said, no, don't put stem cells, stem-like cells. And everybody's putting a stem-like cells now because of that. So they didn't want to accept it. So over a period of time, now everybody's kind of, one of the things I want to tell you, I formed a company called Genostem to develop that P38 inhibitor. The guy who was serving on the board, our company board, he couldn't comprehend that stem cells are 0.5 to 1% of the population. And if you give the drug which targets stem cells, you will not see shrinkage of the tumor. I'm sure we all can comprehend that, but he couldn't comprehend it. If it's a drug, means it has to shrink a tumor. If it cannot shrink a tumor, it's not a drug. I spent 2 years explaining to him. At the end, he said, no, sorry, not interested. Finally, I'm hoping the woman who was going to invest money is coming this week, tomorrow, day after tomorrow. I would be able to convince her now at least. Right? So it's people, they have to really kind of comprehend this. It's a complex. I agree with you, completely agree with you. What we are doing is not even like a 1% of what we should be doing.
Can you re-explain the relationship between ESRP1 and Lumentin? Like, how do they work together?
Great question. So ESRP1 is expressed in an epithelial cells. Okay. So when ESRP1 is expressed, it prevents the cells from becoming a stem cells in response to activation of EMT. Okay. But once a cell undergo EMT, forcefully, whatever method they undergo EMT, on a natural scenario, they lose ESRP1, they gain Vimentin. Okay, they become inversely correlated. So you lose ESRP1, you gain Vimentin. But what Petra found, that if you block cell division and you induce EMT, you can have a Vimentin and ESRP1. Okay, only when you block cell division. Under that scenario, you generate mesenchymal cells, but no stem cells. So in order for cells to become stem cells, they need to gain Vimentin and they need to lose ESRP. So does ESRP1 directly regulate Vimentin? I don't think so. Because ESRP1 is a splicing protein. It does splicing of various proteins. Those splice isoforms do various function. Thank you. Anything else?
So,
you can write to me, and my email ID is just mani@brown.edu. We are also social media, and we post every week. We have a journal club, and the journal club is a little different than everything what you must have seen. Like the student have to present, or whoever, need not be student, whoever presents 5 minutes to summarize the whole paper. Okay, one slide, and then 5 minutes for a discussion. And in a 1.5 hour, we get to discuss nearly 5, 6, 7 papers. Okay, if any of you want to join, send a message to Nick, and he will add you in. And it's very educational kind of a learning platform. You're all welcome to participate. And starting last week, Nick has also started posting them in a social media, like in the summary, what we prepare, we post them in a social media, so just everybody can benefit from it. Yeah. So thank you. I didn't ask whether anyone, there's somebody has a question in a Zoom, I guess. Thank you. Yeah. It's fine.
Yeah.